Good morning, all. Today's my topic is about structure of muscle and muscle proteins. First, muscle mass or muscle tissue. Muscle tissue is made up of a large number of individual muscle cells or myocytes. The muscle cells are commonly called as muscle fibers because these cells are long and slender in appearance. Skeletal muscle fibers are multinucleated and are arranged in parallel to one another with some connective tissue in between. Muscle tissue is separated from the neighboring tissues by a thick fibrous tissue layer known as fascia, which allows the free movement of muscle. The group of muscle fibers called muscle bundles or fasciculi. The three connective tissues were epimysium, which covers the outermost layer of the muscle, perimysium covers each fasciculus, endomysium covers each muscle fiber. Muscle fiber, cylindrical in shape, average length about 3 cm and diameter is 10 to 100 micrometer. Attached to connective tissue called tendon, which in turn attached to bone. Tendon of some muscle are flat and stretched. Such type of tendon called aponeurosis, covered by cell membrane called plasma membrane or sarcolemma. Cytoplasm of the muscle is sarcoplasm. Sarcoplasm contains some glycogen used for synthesis of ATP and also contains red colored protein called myoglobin. MYOFIBRIL, fine parallel filament present in the sarcoplasm of muscle cell. Diameter 0.2 to 2 mu. Length 1 to 4 cm depends upon muscle fiber. Each myofibril consists of two bands, light band or I band, dark band or A band. Light band or I band. It is called isotropic band because it is isotropic to polarized light. That is, when the light is passed, the light rays reflected at same angle. I band is divided into two portions by Z line or Z disk, which is narrow and dark line. Dark band or A band. It is also known as anisotropic band because it is anisotropic to polarized light. That is, when light is passed, the light rays refracted at different angles. This is the pictorial representation of structure of sarcomere. Sarcomere, structural and functional unit of skeletal muscle. Each sarcomere extends between two Z lines of myofibril. When the muscle is in relaxed, the average length of each sarcomere is 2 to 3 mu. Each myofibril consists of an alternative dark and light band. In the middle of the dark band, there is a light area called head zone or Hensen's disk. In the middle of the head zone lies the middle of the myosin filament. This is called M line, which is formed by myosin binding proteins. Sarcomere consists of many thread-like structures called myofilaments. Myofilaments are of two types: actin filament, myosin filament. Actin filament. It is a thin filament, length 1 mu and diameter is 20 angstrom. Extends from either side of Z lines and run across I band and enter into A band up to head zone. Myosin filament. It is a thick filament, length 1.5 mu and diameter 115 angstrom. Situated in A band. Contractile proteins of muscles. The main four proteins involved in contractile elements of the muscle are myosin molecule, actin molecule, tropomyosin, and troponin. Myosin molecule. Each myosin molecule has two portions, tail portion and head portion. Tail portion. It is made up of two heavy chains which is twist around each other in the form of double helix. Head portion. At one end of the double helix, both the heavy chains turn away in opposite direction and from the globular head portion. Thus, the head portion has two parts. Each myosin head has two attachment sites. One site is for actin filament and another site is for ATP molecules. Myosin head is absent in the central part of myosin filament, that is in the head zone. Actin molecule. Major constituents of thin actin filament. Each actin filament, F-actin, which is a polymer of small protein G-actin, contains 300 to 400 actin molecule and their molecular weight is 42,000. Actin molecules are arranged in double helix form. It has active site which myosin head is attached. Tropomyosin, present along the double helix strand of actin filament and their molecular weight is 70,000. In relaxed condition of muscle, the tropomyosin molecules cover all the active sites of F-actin molecules. Troponin, formed of three subunits, troponin I, troponin T, troponin C. Troponin I, which is attached to F-actin, troponin T attached to tropomyosin, troponin C attached to calcium ions. Other proteins of the muscle are actin attaches actin filament to Z-line and desmin binds Z-line with sarcolemma, nebulin, which runs in close association with and parallel to actin filament, and titin, large protein connecting M-line and Z-line, dystrophin, connects actin filament to dystroglycan. Thank you, ma'am.